Well, thank you for the opportunity of presenting our data in this session. The work that I will show you is a collaboration between the research laboratory on CLL from the Institut Pasteur de Montevideo in Uruguay and the Feinstein Institute for Medical Research in New York, and I have no disclosures. So, as it was mentioned just a moment ago by Shishi, in order to continue studying the role of AID, this mutagenic enzyme in CLL, we crossed the TCL1 mice with transgenic strains overexpressing AID, and we generated what from now on I will refer to as double transgenic mice. These double transgenic mice with AID overexpression showed shorter survival and increased Ki67 positive cells. So the aim of this specific part of the work that I will show you was to characterize the in vivo AID genome imprint in this progressive CLL model. In other words, said to dissect the AID of target mutational activity. So for that, we first performed a somatic variant calling on whole exome sequencing data obtained from the genomes of purified leukemic cells from 7 double transgenic mice and 5 TCL1 mice. And what we saw by Virtue scan analysis was that while all mutations, overall mutation rate remained unchanged between the two groups of mice, AID-related mutations were increased in double transgenic mice as compared to TCL1 mice. So these AID-related mutations were searched by different approaches. One of them was an unbiased somatic signature discovery analysis in which we uncovered that the higher mutations of AID were due to transitions from C to T at RCY motifs, where R is one of the two purines and Y is one of the two pyrimidines, and that matches the canonical AID signature that has also been mentioned earlier today. So the genes that were mutated in the double transgenic mice only, not in TCL1 mice, by AID and with canonical AID signature, that were mutated 5 times or more in one mouse or some in the 7 double transgenic mice included in the study are depicted in this graphic. We saw 6 genes that have been previously described as AID of target in murine models. We also detected novel mutations in transcription factors related to tumor development, like GATA3 or SOX4. And when we developed a pathway analysis, we saw significant enrichment in these 3 pathways, importantly including Wnt signaling. So, and these 6 genes that are highlighted here in yellow are genes that have been previously described as putative gene drivers for B-cell neoplasms like CLL or diffuse large B-cell lymphoma in different reports. So, as I said before, double transgenic mice show increased levels of Ki67 positive cells, so we took advantage of that, and we purified by cell sorting leukemic cells from spleens of double transgenic mice and TCL1 mice, those Ki67 positive and Ki67 negative, to study AID activity specifically in proliferating cells. And we saw again that the signature that prevailed and explains the differences in the mutations between the two models are the canonical AID signature being increased in the double transgenic mice as compared to TCL1 mice, again in proliferating cells, and that again one of the signaling pathway that is being highly mutated by AID in proliferating cells is the Wnt signaling pathway. And these are the gene drivers included in the list of mutated genes by AID in proliferated leukemic cells. So the last part that I will show you has to do with an attempt to study from the list of mutated genes in our model those that have mutations that coincide exactly with previously described mutations in human tumor samples. And we found that 4 genes that are PIM1, MCL1, and the histones 1E and 1D, the amino acidic changes occur at identical sites in human and in mouse proteins that we detected. And we took advantage of previously crystallized proteins So we model what the mutation would do. For example, for PIM1, the exchange of serine 97 to asparagine will affect the domain that negatively regulates PIM1. So this kinase could be positively hyperactivated by this mutation, and that for the histone 1D, the exchange of the serine 90 would cause a steric hindrance due to clash with the nucleosome DNA, would putatively lead to chromatin decompaction and favoring transcription. So in sum, we showed that canonical AID mutations is the main signature detected, that Wnt signaling pathway is being highly affected by AID, and that previously described human tumor driver genes with identical mutated sites have been detected in this novel murine model, linking AID activity in vivo with tumor clone aggressiveness in CLL. Thank you very much to the group in Uruguay and to the group in the Feinstein Institute.
Thank you, Pablo.